Good evening, everyone. I hope my voice is audible and people are still joining the session. So let's just wait for 5 minutes and then we'll begin with this. All right, so welcome everyone to another live session of Basics of Crop Breeding and Plant Biotechnology, Week 4. I am Prashasti Pandey, NPTEL PMRF, PhD Scholar, Department of Botany, Banaras Hindu University. And if you all have attended previous sessions, in these live sessions, we will be going through some questions from previous year's assignments, and we will be solving them with the concept of the question. And hopefully, these will help you in solving this year's assignment as a part of your course. Okay, so what we will be doing is we'll be going through, I'll be giving one question on screen. I'll give 2 minutes of time. If anyone knows the answer, you can either write it in the chat box or you can tell the answer aloud, and then we can start discussing the answer. Okay, so I hope this is clear. Let's move ahead with our first question. Okay, so the first question is choose the correct option for mass selection. The options are mass selected variety have narrow genetic base. Adaptability is more in self pollinated variety than cross pollinated variety. In the case of self pollinated crops, a mass selected variety is heterozygous and homozygous in nature. And in the case of cross pollinated crops, mass selected variety is mixture of homozygous and heterozygous in nature. Okay, I'll give you 2 minutes if anyone knows the answer. Okay, so somebody has written answer as option D. Okay, so let's solve this. All right. So first, although we have done this topic a lot of times before, but let's try to understand what mass selection process is first. So basically, mass selection is the process of selecting many phenotypically superior plants. Okay. So this means that the process of selection in case of mass selection is totally dependent on the phenotypes of the plants. It has nothing to do with the molecular combinations or anything like that, and it will basically, okay, just give me a second. Okay. So it basically means that because the plants are phenotypically superior, the traits that we require. So phenotype basically means the trait that we actually require. That means the grain size will be larger, the amount of grains will be more, the amount of pods will be more. So these kind of things come under phenotypes. Okay, so the plants which are superior phenotypically, they will have better phenotypes of that particular plant. Okay, so mass selection will totally depend on selecting those plants which are phenotypically superior, and then after their selection, their seeds are basically bulked and developed into an improved variety. Okay, so there is no basis of selection on no molecular basis of selection of these plants, whether the plants are heterozygous or whether they are homozygous. We have no idea about that. Just because the phenotypes are better than other plants, they are selected. Okay, so if you see in the figure also, in the first year, the large number of phenotypically similar plants or superior. So similar here word is used because most of the plants are superior and that's why they represent similar traits. Okay, so they are selected on the basis of the vigor, the plant type, and disease resistance. And then plants from them are selected, and the seeds are then composited and raised in next generation. Now, in second year, these composite seeds are planted in preliminary yield trial along with standard check variety. Now, the variety of the selection will be included, such as like checks to determine if there is any improvement in the selection, if any variety comes out as better than the than other all of it, and then they are further selected. And then this variety is then evaluated in coordinated yield trials and in several different locations for different traits that we are trying to select. Okay, by the end of sixth year, this seed multiplication becomes enough for distribution, and then it is labeled as a new and improved variety. Okay, so this is again mention the same thing here, that the steps will be that a genetically variable population is first grown, and then many superior plants are selected on desirable traits like height, yield, and disease resistance. And then the seeds are basically harvested from selected plants. They are bulked and mixed together, and then basically the sown seeds can produce improved variety after growing multiple generations, after going through multiple selection procedures at different locations and different after different trials done on them. So now the key characteristic features of mass selection is that it is based on phenotype. Okay, this is the most important thing in case of mass selection, that it is not based on genotype. Only phenotype is considered for mass selection, and seeds of these plants are basically mixed together because there is no concept of genotype. That means whether the seeds are heterozygous, homozygous, or their genetic combination is not considered. Therefore, whatever plants are of good quality, their seeds are collected and mixed together. Now, because the genetic combination is not taken into consideration, the population of the seeds, and also they are mixed. Therefore, the population is usually heterozygous. Okay, there's very less chance that homozygous population is there because we are not crossing them. We are not generating some seeds to make the genetic combination as same. So most of the seeds are heterozygous. And now the most important thing, that mass selection is basically used in cross-pollinated crops, because heterozygosity can only be maintained in cross-pollinated crops. When the crops are self-pollinated, that will result into higher and higher population of homozygous varieties. Okay. So, in case of self-pollinated crop, there are more chances of homozygous varieties. But in case of mass selection, there is more chance that the population is heterozygous, and therefore it is more preferred in cross-pollinated crops. In certain cases, it can also be used in self-pollinated crops. But eventually, the population will become homozygous, and the variety which is there will keep on reducing in case of self-pollinated crops. Okay. Now, what is the advantages and what is the disadvantages of this particular population? This particular selection method. Okay. So basically, the advantages, in terms of advantages, this method of mass selection is basically very simple and it is inexpensive. Okay. Also, it will maintain the genetic variability, because again, we talked about the homozygosity. So, genetic variability is maintained. Overall population in this case is improved for performance, and it is useful for improving local varieties. But the problem with mass selection is that it will be less uniform than compared to the pure line selection. Because the population is heterozygous, we don't know the seeds that we have achieved were showing a good quality in the parent, but that doesn't certainly mean that when the next generation is grown, it will also have good quality. Okay, so it will be less popular. The chances of selection, the chances of offsprings that we have collected will not be as uniform as we attain from pure line selection. Also, the improvement is very limited, because the traits will ultimately come from the population that we are picking. We are not adding any other trait from some other population. So the improvement will be limited to the traits of that particular population only. Third is that selection is very less precise and it is based only on phenotype. That is why there is no role of any molecular advancement in this case. Okay. Now, based on this, let's talk about the answer. The correct option for mass selection will be D, that in the case of cross-pollinated crops, mass selected variety is mixture of homozygous and heterozygous. Okay, this was the last line. It cannot be C, because in case of self-pollinated crops, mass selected variety is heterozygous. No, it will not be heterozygous. In case of self-pollinated crop, the mass selected variety will only be homozygous in nature. Okay. Second option was adaptability is more in self-pollinated variety than cross-pollinated variety. No, it has basically nothing to do with adaptability. And also, if we talk in terms of adaptability and not mass selection, adaptability will be more in cross-pollinated variety. Because in case of cross-pollinated variety, the chances of survival, because it comes from mixed population, it gives more chances of survival than in case of self-pollinated variety. And the first option was mass selected variety have narrow genetic base. No, it will have a very broad genetic basis, because the selection was not based on any genetics. The selection was based on only phenotype. That means 10 different genotypes can also generate one single phenotype. And one single phenotype, because it is backed up by so many genetic bases, the genetic base for mass selected varieties will be huge. So first option is also wrong. So only D option is correct. Okay. So that is that in case of cross pollinated, the selected varieties will have both homozygous and heterozygous in nature. Okay. So let's move on to our next question. With reference to mass selection of crop plants, consider the following statements. Mass selection has been more commonly used for the improvement of cross pollinated crops than single plant selection. B. The purpose of mass selection is to increase the frequency of superior genotypes in population. C. In this method, the individual plants are selected on the basis of observable genotypic traits. And D. This method of breeding has been very effective in improving characters governed by many genes. Which of the following statements are correct? Now, this is an MSQ question. That means more than one option can be correct. If you know the answer, you can tell it. I'll give you 2 minutes. Okay, we have two answers. One is A, one is D. Anyone else? Okay, so let's discuss this answer. So we have in the previous question discussed a lot about mass selection. So let's just understand all the statements and then see if they are correct or wrong. So the first statement was that mass selection has been more commonly used for the improvement of cross-pollinated crops than single plant selection. Yes, so the statement is correct because we discussed in the last point that mass selection is very useful for cross-pollinated crops, and especially cross-pollinated because it maintains the genetic variability. Okay. Second one is the line talks about single plant selection. So single plant selection is also called as pure line selection. So this is basically more effective for the self-pollinated crop. Okay, I have discussed this before also. It is more useful for self-pollinated crops. Okay, so mass selection is more useful for cross-pollinated, and for self-pollinated crops, single plant selection or pure line selection is useful. So option A is correct. Option B. The purpose of mass selection is to increase the frequency of superior genotypes in the population. So, yes, this is correct. Okay. So, yes, this statement is also correct because in case of mass selection, it will work by selecting the superior plants and then bulking their seeds. Okay. So, this also increases the frequency of desirable alleles or desirable genotypes in the population. Although the selection is not based on this, but once we keep on selecting the plants with better phenotypes, they will also have better genotypes. So, yes, the mass selection will also increase the frequency of the superior genotypes in the population. The statement is also correct. Now, option C, that in this method, the individual plants are selected on the basis of observable genotypic traits. No, we have talked about this that in mass selection, genotype traits are not considered in any case. Okay, they are based on phenotypic traits, which are observable characters. Okay, the ones which we can see in the plants. It has in genotypic traits are not observable. So yes, the selection is based on phenotypes, and genotypes cannot directly be observed in mass selection. Of course, it at the back end, the genotypic traits are increasing. The better genotypic traits are only carry forwarded, but they are not considered. So the correct statement, if in this case, would should be, instead of genotypic traits, it should be phenotypic traits here. Then the statement would have been correct. But for now, it is wrong. Then the statement D was that this method of breeding has very effective for improving traits that are governed by multiple genes. Yes, because when the traits are governed by multiple genes, you cannot select plants on the base of single genes. Okay, so suppose there is some trait which has four genes controlling it. Now, if you improve one gene, then first of all, it is very difficult to improve or incorporate four higher quality genes in a plant. So it becomes very difficult to control that particular trait. But now, when you get the seeds of the seeds from a population, and you see the phenotype that you require, no matter how many number of genes are controlling it, you are selecting the one which has better phenotype. So ultimately, at the end of many populations, you will have the plants with the better quality phenotype, regardless of what genotypic traits they have. So, yes, mass selective is very effective for quantitative traits, also called as polygenic traits, like yield, like height, adaptability, stress tolerance. All these traits are basically polygenic traits. That means they require multiple genes to control them, and that is why it becomes easier for their selection through mass selection procedure. So, in this case, option A, B, and D were correct. Yes, A and D were right. Also, option B was also correct. Okay, let's move on to next question. Half sib method is modification of which breeding method? Options are pedigree selection, recurrent selection, bulk method, and mass selection. Anybody knows the answer to this? Okay, we have one option B. Anyone else? Okay. So let's discuss this. First, let's talk about what is half sib method. So half sib is basically short form for half sibling. Okay. So in half sibling method, basically what happens is, so there will be one common parent. Okay. So usually the female in this case will be common, and there will be different pollen partners. Okay. So a female is basically selected of a superior variety, and then it is crossed with different pollens to get different results. Okay. So in this case, 50% genes will be from one single parent, and the other 50% will come from different parents, and that will produce a different types of recombinations of the gene, and then the best one out of it is selected. So basically, half sib method is modified form of mass selection because we don't know the genotypes, but we keep on changing the male parent so that in expectation with that the outcome from them will give some of the superior kind of phenotypes, and then we will simply select them and go on with the procedure of mass selection. Okay. And it is also done and used in many cross pollinated crops. So why how it is related to mass selection? So in this case also, basically a superior plant is selected on the basis of phenotypes only, and then the seeds are bulked. But in case of half sib method, basically selection is based on superior plants, but the progeny testing is done on half sib families because, see, like this is the common female. Okay. And now it is crossed with different males. Okay. So now the offsprings that come from all of this will become half siblings because they will share 50% of their genetic information altogether, because that is coming from their mother. Okay. Only their 50% genetic combination will be different. So, in this method, again, the selection, the seeds, the outcome will be based on phenotypic traits only. And these phenotypic traits will help in selection. But we are trying to introduce some recombination, some heterozygosity in order to achieve a better phenotypic trait by changing the male parent and keeping the superior female as constant. Okay, so this basically becomes one step ahead of mass selection in case of precision. Okay, in terms of precision, it becomes one step ahead of mass selection. Now, this is explained in the simple figure only. Like there is a field population and then there are unselected plants. Now, in case of open population, the seeds are collected from the selected plants. And now, the selected plants, there will be common female parent and different male parents are crossed. And this progeny will be grown from each selected parent. Okay. So now half of the siblings are evaluated, and then they are selected for next generation, and then that is how an improved variety is generated. Now, why it is considered as a modification? Because in both these methods, the superior plants form a population, and they come from population. They are selected from a population. Also, this method basically adds progeny testing, so it will improve the selection accuracy, and it is also called mass selection with progeny test. Okay, so now coming back to the question that half sib method is modification of which breeding method? Option D is correct, that it is the modification of mass selection method. Okay, okay, let's move on to our next question. Tester used in RSSCA is having which of the following condition? Broad genetic base, inbreds, low yield potential, and narrow genetic base. Okay, someone has written as answer D. Anyone else? Okay, so let's discuss this answer. So first of all, what what is the term RSSCA? Okay. RSSCA is basically short form for reciprocal recurrent selection for specific combination ability. Okay. So it is basically a breeding method in which two populations are simultaneously improved. And this is done by selecting individuals based on their cross performance with opposite population and then using narrow based testers, okay, which are usually inbred lines. Now, what is the purpose of doing it? First, the purpose is to improve the specific combining ability. Okay, that the plants are able to combine with other populations to improve this. This particular method is done. Then, because they are able to combine, there are higher chances to develop a superior hybrid because of that. Okay, and the third is that it is basically more useful in cross pollinated crops like maize and pearl millet. Now, what are the key features of this RSSCM? So it will involve two populations. Okay, as the definition says that it will basically help in the improvement of two populations simultaneously. Okay, so two populations here in this will be involved, and then the selection will be based on cross performance. Inbred testers, which are narrow genetic base, are used in this, and this will improve both populations but reciprocably. Now, um, okay. So, what are the steps involved in this case? So, basically, first, the plants are selected from population A. Okay. And then they are crossed with basically population B. They are crossed with the tester from population B. And then the best performing cross will be selected. And then that selected cross will be intermate with the population A. Simultaneously, the plants will be selected from population B also. And from population B, selected plants will be crossed from the tester from population A. And then there also the best crosses will be selected and then intermated with the plants from the population B. Okay, so this cycle, when it is repeated in both the populations, it will result in improved varieties of both the populations simultaneously. Okay, so this is basically the method of recurrent recurrent reciprocal selection for specific combining ability. And in this case, the tester that is used is an inbred line. Now, why inbred lines are used as testers? The reason first is that inbreds are basically genetically uniform varieties. Okay, so they will usually be homozygous, and this will uh, give a better chance for the combination to be same. Okay, so for example, that the inbred variety has this as their genetic trait. Okay, so no matter which of this allele goes, the offspring will always receive only one type of allele. So this will give a better uniform, genetically uniform offsprings, and it will be homozygous. Second, they have narrow genetic base. There is less chance of variations produced in the offsprings. Third will be that this will allow accurate evaluation of specific combining ability because there can be no doubt that either one of the allele is combining more or the other is combining less. No, because the population is homozygous, it will give equal chances of the alleles to combine, and the evaluation will be accurate on the basis of combining ability. And then fourth will be it will help identify the individuals that perform the best specific crosses. Okay, so based on this, the question was tester user is having which of the following condition? Option B is correct that the tester users are inbred so that they will have homozygous genetic combination and it will be more helpful in determining the specific combining ability. Okay. So the next question is if the arbitrary value of parent A is 30 and parent B is 20, then what will be the values of F1 showing heterobeltiosis, complete dominance, and partial dominance? I give you 2 minutes. You can try solving this question. Okay, so let's solve this. So what is given in the question, that parent A, the value for parent A is 30 and for parent B is 20. Okay. So now let's first calculate the mid parent value. So mid parent value is nothing but the average of two values. So it will be 30 plus 20 divided by 2, which is 50 by 2. So the answer is 25. Okay. Now we have three conditions. The question is talking about three conditions. One is heterobeltiosis, complete dominance, and partial dominance. Now, in case of heterobeltiosis, basically the resulting population or F1 progeny will be better than the better parent. Okay, so better than the better parent means if parent A as value is 30, that means the F1 must be more than 30. Okay, because it will perform better than the better parent. So the value in case of heterobeltiosis, the value will be more than 30. In case of complete dominance, the value will be the F1 resulting population's value will be equal to the better parent. That means F1 will be equal to 30. And in case of partial dominance, it will be less than the better parent but more than the mid parent value. Okay, so that means it will be somewhere between 25 to 30. That means in first case, the value should be more than 30. The second case value should be exactly 30, and in third case it will be somewhere between 25 to 30. Okay. Now based on this, let's check our options. So in option A, it was 37, 29, and 19. So this cannot be correct because 37 is more than 30. That is true for heterobeltiosis, but 29, but second has to be 30. So that is wrong. And 19 is less than 25. So that will also be wrong. In option B, 20, 20, and 10, all are incorrect because this is not more than 30, this is not equal to 30, and this is not between 25 to 30. Option C, 40 is more than 30, but 35 is not equal to 30, and 28 is also right, but of course, this one is wrong. So that means this option is also wrong. But in case of option D, so 50 is more than 30. That means heterobeltiosis is correct. 30 is happens in complete dominance equals to better parent. That is also correct. And 28 lies between 25 to 30. That means partial dominance is also correct. So in this case, option D is correct. Okay. The answer is 50, 30, and 28. I hope this question was clear. Let's move on to next question. Choose the incorrect statement for the modification of mass selection with progeny testing. The option is it combines mass selection with evaluation of progeny to improve the efficiency and precision of the breeding process. It helps to preserve the genetic potential of selected parents and the ability to pass desired traits to offsprings. Superior plants are selected in the early stages while inferior plants are removed. It is often used to evaluate performance of progeny to ensure the selection of plants with stable and heritable traits. So let's solve this question. Okay. So a modification of basically mass selection with progeny testing. It is also called as half-step selection, which we have discussed earlier. So it will involve selecting plants and then evaluating their progeny to confirm the genetic superiority. Okay. Now, let's talk about the statements. So, the first statement was that it combines mass selection with evaluation of progeny to improve efficiency and precision of breeding process. Yes, it is correct. This is basically the main purpose of the modification of mass selection. Then, progeny testing will increase the accuracy compared to the simple mass selection. Yes, of course, this is exactly what we are talking about in half SIP case that. The progeny testing will be one step ahead of the simple mass selection. So yes, option A is correct. Then option B. Uh, okay. So option B is it helps to preserve the genetic potential of selected parents and their ability to pass desired trait to offspring. Yes, this statement is also correct because in case of progeny testing, it will confirm whether the desirable traits are heritable or not, and it will ensure that the selected parents truly have good genetic value, which does not happen in mass selection only. Then statement C was superior plants are selected in early stages while inferior parents are removed out. No, this is not correct. It basically this describes simple mass selection method and it is not a modified method. In the modified method, the progeny evaluation is essential and only then we can select the parents and it is not an early phenotypic selection or removing of any plants. So this statement is correct. Option D is it is often this statement is wrong. I'm sorry, this statement is wrong. Option D was it is often used to evaluate performance of progeny to ensure the selection of parents with stable and heritable traits. Yes, of course, this is the core feature of progeny testing that it only talks about the heritable traits and it will select the parents based on ensurety that the traits are stable and heritable and it is genetically controlled and stable. So based on this, option C is the incorrect one, that superior plants are selected in early stages. It is nothing like that in case of selection. It requires some time, and the plants are not removed or selected based on any superiority or inferiority in early stages. Okay, so next question. Which type of population is improved in reciprocal recurrent selection? The option is single population is selected for desirable traits, two populations are improved simultaneously, two an inbred line is used for improvement, and two homozygous populations are used simultaneously. Okay, so let's discuss this answer. So we have discussed this earlier also that reciprocal recurrence selection is basically a breeding method. It's a breeding method that mainly uses cross-pollinated crop to improve two populations at the same time. Okay, for their combining ability. Now we have learned this that how it works. That population A plants are crossed with testers from population B, and population B plants are crossed with testers from population A. And then best performing crosses are identified. Selected plants are intermated with their own population, and this will help improve the population. This will help improve the crops from both the populations simultaneously. Now, let's talk about the wrong options. That a single population is selected for desirable trait. No, this is because this describes a recurrent selection. It is not a reciprocal recurrent selection. In this, single trait is because we are talking about two populations. A single desirable trait is not selected. Then an inbred line is used for improvement. Basically, RRS uses population, not a single inbred lines. Okay. So it will not use single inbred line, and populations are used in this. So this option is also wrong. Two homozygous populations are used simultaneously. No, two heterozygous populations are basically used, and there is no homozygous population in this. So option D is also wrong. So option B is correct in this case, that two populations are improved simultaneously. This is reciprocal recurrent selection. Okay. Okay. So our next question is, which method is used to increase the homozygosity? Recurrent selection, mass selection, inbreeding, and hybridization. This is very easy question. Okay. So let's talk about the answer. So two people have answered as option C. So it is very simple actually. Basically, inbreeding is the answer because inbreeding in this method it increases the homozygosity in the population. Now, why inbreeding? Because it involves mating between the genetically related individuals. Okay, like self pollination crops. So, of course, because they're genetically related, this will increase homozygosity in the population. It will reduce heterozygosity generation after generation, and it is used to develop pure lines, or they're also called as inbred lines. Okay. So, what happens during inbreeding is that basically homozygosity will increase by 50% in each generation, okay, in self pollinated crops, and eventually in one population will come where the crops are completely homozygous pure lines. Now, why the other options are incorrect? Because in case of recurrent selection, it is used for improving population. It maintains the variability. Heterozygosity is the main source, and it will specifically not increase the homozygosity. Mass selection is completely based on phenotype. It has nothing to do with heterozygosity or homozygosity. It will only it there is no definite means to say whether homozygosity is increasing or heterozygosity is increasing. And as case of hybridization, the basic concept is increasing heterozygosity and not homozygosity. Okay, so yes, option B was correct. Option C was correct. Inbreeding is used to increase the homozygosity. Now the next question is: open pollinated variety can be associated with which of the following in breeding methods? And this is the MSQ type question, so multiple answers can be correct. Options are pure line selection, pedigree selection, recurrent selection, and mass selection. Okay, so somebody has said B and C, and someone has said C and D. Okay, so let's discuss this. So, an open pollinated variety. So, basically, an open pollinated variety is genetically variable population that is maintained by open population and natural crossing. Uh, Basically, OPVs are usually associated with main breeding method, which is to maintain the genetic variability. And this is normal type of procedure where the plants are let to be pollinated um, by the whatever the pollens that are available, and natural crossing happens. In this case, pure line selection is incorrect because pure line selection requires genetic and uniform and most importantly homozygous lines, which is not possible in case of open population and natural crossing. So uh, because this will always produce more and more heterozygous varieties. So it cannot be close to pure lines. Then, in case of pedigree selection will mostly use self-pollinated crops, and it will lead to more pure and uniform varieties, but not in case of OPVs because there will be more heterozygous varieties. Then C is recurrent selection. It will use mainly cross-pollinated crops, so it is correct in OPVs that more type of cross-pollination will happen and it will maintain the genetic variability, which will also improve. It is improved pollinated population. So recurrent selection is correct. Mass selection, yes, mass selection will also be correct because it is the most obvious way of OPVs that the plants are led to be pollinated and the seeds are just selected on the basis of phenotypes and they are bulked more and more. So it will maintain the heterozygous population structure. So in this case, option C and D is correct that OPVs are associated with recurrent selection and mass selection. Okay. So the last question is match the following. In column A, you have simple recurrent selection for specific combination combining ability, recurrent selection for general combining ability. And column B is non-additive gene action, test cross performance, and based on phenotype. We have discussed all these three. I hope it will be easy for you to solve. I'll give you 2 minutes. Please try solving it. Okay, so we have a lot of different answers here. So let's discuss this. Okay, so let's go one by one. First was simple recurrent selection. Now, simple recurrent selection is based on phenotype, it is based on observable traits, and no tester is used. So it matches with based on phenotype. Then, in case of recurrent selection for specific combining ability, it will basically improve the specific combining ability and it is associated with non-additive gene effect. Okay, so it will match with non-additive gene action. Okay. Next is recurrent selection for general combining ability. So basically, in this, tester with broad genetic basis is used, and selection is based on test cross performance, and it will improve the additive gene action, that is GCA. So it will match with test cross performance. So option D is correct. That your one will match with C. That simple recurrent selection is based on phenotype. Your second will match with A, that RSSCA is based on non-additive gene function, and your RSGCA is based on test cost performance. So option D is correct. Okay, I hope all these questions were helpful and you were able to understand all of them. If there is no doubt, we can end this session and we'll meet again in next session next Tuesday, same time.